In this video, we are going to be looking at the different steps in translation for prokaryotes. When it comes to translation, we have several molecules that are needed in order for the process to translation to occur. We have introduced throughout this chapter each of them, but let's just review. So, in order for translation to occur, we need a tRNA molecule, and the role of the tRNA is to bring the amino acids. The ribosome, which contains a large subunit and a small subunit, is going to be the site of protein synthesis. That's where we're going to be making the amide bonds. We also need mRNA, and remember that the mRNA is going to be the template for protein synthesis. In this figure, we are not illustrating the ribosomal RNA, but understand that ribosomal RNA is also involved. It's not illustrated. And is located at the ribosome. So far, we already have discussed in previous chapters the process of DNA replication, the process of transcription. And in translation, we see that similarly to the other processes, we're also going to have three phases. Here we're going to have similarly an initiation phase, an elongation phase, followed by a termination phase. Let's get started explaining what is occurring at the initiation phase. So, what happens in the initiation phase is that we are going to have an initiation complex. Understand that this initiation complex is going to have the FMET tRNA FMET, which in a way, this is going to be the formal MET tRNA. We're going to have the identification of an initiation codon. In the mRNA, we're going to have the small subunit and the large subunit of the ribosome come together. We're going to have some initiation factors, and as you can see, they are designated by the numbers 1, 2, and 3. And lastly, we're going to have GTP and magnesium present. So let's explore what happens during this stage. So I'm going to be focusing on the left side of the slide, which is where we have the figure for the initiation step. So what is occurring here is that we have our mRNA template. And again, remember that the transcription that we're talking about is the one in prokaryotes. So this is not modified like we saw in eukaryotes. Okay? So we're going to have a small subunit of the ribosome come and interact specifically with our mRNA template. Now, as you can see, during this process, we need GTP, we need initiation factor 2 that is going to be interacting with the F-met tRNA F-met, okay? And again, this molecule is the N-formal methionine, and they are going to come together in the following manner. When they are interacting, understand that it is at the Shinedalgarno sequence where the binding is going to be occurring for the ribosome to identify that that is the site for protein synthesis. At the template, the AUG site, which I'm going to highlight in green, is identified by that N-formal methionine. And as you can see, the anticodon, let me just highlight the anticodon. The anticodon in the formal methionine is going to be UAC, okay? And as I mentioned before, this is where we have the first amino acid in the protein synthesis. Then, on the second step, as you can see, we're going to have the initiation factor to leave. We're going to have the large subunit bind where we have the small subunit, and we already have the N-formal group identifying that start site. And as you can see, in addition to the IF2 leaving, we also have hydrolysis of GTP, because as you can see here, we started with GTP. At the end of this, and the beginning of that second step, we have the release of GDP plus phosphate. And we also have, as you can see, the release of initiation factor 2 and initiation factor 3. So by the end of the initiation phase, now we have the large subunit of the ribosome, the small subunit of the ribosome, bonded to our template, which is the mRNA structure, okay? And we also have already the first amino acid in the sequence, which is going to be that formal and methionine. And specifically, this is going to be bonded to the P site. We also have an A site and an E site. And we're going to see their roles in the slide when we talk about elongation. So, to talk more specifically of what is a Shinedalgarno sequence, as you can see, this is going to be the ribosome binding site in bacteria. And it is about 10 nucleotides upstream of the start codon. So that means that on average, this is going to be negative 10 base pairs. This region is going to be conserved throughout evolution. So that's why you see this blue region identifying that if we look at different bacteria, is going to have this Shinedalgarno sequence. And another thing that I should point out is that we also have the initiation codon that for most organisms in those species is going to be given by the nitrogenous bases AUG. Remember that we read them in 3 letters, so this is called the start codon. Let's continue talking about elongation phase during translation. So as I mentioned before, once we have this large subunit and small subunit bonded to our mRNA, and we already have that first tRNA with the amino acid present, let's establish that in specifically the ribosome unit, we're going to have 3 specific sites in which chemistry will be occurring. More specifically, when it comes to the important sites, we have the A site, which is called the amino acyl site, the P site, which is called the peptidyl site, and this is not illustrated, but I'm just going to write it in. This is the E site. This is going to be the exit site. So let me just write them in the corner. So we have A site, this is an amino acyl site. We have the P site, which is the peptidyl site. And we have the E site, which is the exit site. So the first thing is that we should be reminded that when it comes to reading the mRNA, we're doing in 3 letter sequences. Remember, we're going to be reading the codons. The tRNA molecule is going to have an anticodon. Okay? And this anticodon is going to be complementary to the codon. Okay? So what determines the sequence of amino acids that we are going to have in our peptide is the sequence on the mRNA. So the amino acid that is incorporated onto the protein is determined by the codon on the mRNA molecule. The reason why I specify that is because some students assume that the anticodon is what matches the amino acid. No. The amino acid synthetase that makes those tRNAs has a very tough job because it needs to know the amino acid that the anticodon is going to be complementary with in order to attach the amino acid. So on step 2, once the codon has been recognized, we're going to see that between the A site and the P site, we are going to be forming the peptide bond. Then the growing protein chain is transferred from the P site to the A site, okay? So now we're going to have the growing chain on the tRNA that is on the A site. And then as you can see, we're going to have hydrolysis of GTP. And after this hydrolysis of GTP, we're going to have translocation of the free tRNA that was in the P onto the E site so it can exit. And we're going to have the tRNA that now has the growing chain of polypeptide move onto the P site. So now another 3-letter codon can be identified by a tRNA, and we continue growing the peptide chain or the protein chain. So let's look specifically at what is happening in between the P site and the A site. As I already established to you guys, A site means amino acyl site. P means peptidyl site. So what happens overall in the ribosome, so this is kind of like a snapshot. At the ribosome, P site and A site. As you can see, in order for making that peptide bond, that amide bond between the two amino acids, we have the amino group of the amino acid that it is on the A site do a nucleophilic attack on the carbon that is part of the peptidyl chain. So that carbon is going to be our electrophile. As you can see here, we also have the rRNA being involved in this process, okay? Which in this case, as you can see here, what is occurring is that is going to be deprotonating the amino group on the amino acid on the A site. And in the ribosome, as you can see, that's how now we have a new peptide bond, and we have now the transfer, okay, of that new protein chain that is now located on the A site. Now this is a free tRNA that will move to the E site to exit the ribosome, then to get activated and come in if it's necessary. And then remember that overall, that now tRNA that is on the A site, and it has the new protein chain because we incorporated one new amino acid, is going to move to the P site to allow for the next codon on the A site to be read. And that's how it grows. Now, when it comes to the termination of translation, as you can see, once our protein, it is on the P site, once we have reached a stop codon, what happens is that now we have a release factor that is going to bind to the stop codon. So as I mentioned to you guys, stop codons do not encode for an amino acid. Their role is simply having the release factor bind there, so the ribosome, the tRNA, and the free polypeptide, in other words, our protein of interest, is now released into the cytosol. And overall, what's going to happen is that then that whole amino acid chain is going to be undergoing the folding for generating the secondary structure, tertiary structure, and quaternary structure if it contains it.